Okay, welcome to 10C, our third and final installment of Lecture 10, which is part 2 of Vision Neurophysiology. This last part is all about perception, so that it's the conscious awareness and interpretation of some sensory information, in this case, visual information. First, let's remind ourselves how we got here. So all of that stuff that you learned about the retina and then the signals traveled from the retina through let me pick a different color, blue. All right, through the optic nerve, and then they either stayed on the same side at the optic chiasm, went through the optic tracts to the lateral geniculate nucleus, and then through the optic radiations and back to the visual cortex. Or they actually, I'll switch my color here. If they were coming from the nasal side, then they actually crossed at the optic chiasm through the optic tract to the lateral geniculate nucleus of the thalamus, then through the optic radiations to V1, or the primary visual cortex, which we find in the occipital lobes. So our signals arrive here at neurons in the cerebral cortex at V1, this primary visual area. So this is a diagram from the LIM textbook, and what they're showing you up above is the visual field. So, and where everything then maps onto is going to map onto the retina and then onto the different sides of the brain. So we've got information from here, for example, ending up over here on the left side of the brain, because that was from the right visual field. And we've got information from here ending up over here. So you can see all of those differences. Now here they've used red and blue in a different way. So the red corresponds to things that are from the macula. So that area where we have the highest concentration of photoreceptors, particularly cones. So we get a lot of granular information. So notice that even though it's like less than 5% of the overall visual field, look at how much brain area is designated to processing this information, right? This is where the money is. This is where we're spending a lot of our energy kind of trying to interpret the things that we're looking directly at, which makes sense. You'll also see how things that are adjacent to each other in real life are kind of adjacent to each other in areas of the brain. We'll see that kind of regionality when we study the parietal lobe and the somatosensory cortex. It just kind of makes sense. Things just kind of follow similar patterns. So we're arriving here in the cerebral cortex in the occipital lobe at V1, the primary visual area. And so those are the same thing. V1 is the primary visual area. And what we're getting here is raw data. So I'm seeing a bit of blue over here and then an area of dull pink over there, right? So just the raw data of what colors, the brightness, how much of it, how little of it, what areas of the retina are detecting it. So this is raw data. So this small bit of blue over there and large area of dull pink over here could actually be a baby's face, right? But at this point, we just know there's some blue and there's some pink and there's a little bit of green up in the corner, right? And this part of the brain doesn't know what it is, but it just says, this is the information I'm getting. So as you look at this particular image, there's a bunch of black area and there are some white areas. And then think about what you see when you look at that. If I asked you to tell me what this is a picture of, some of you will tell me that this is a picture of like an urn or a vase. If you are kind of picking up on the black, right? So this is kind of the outline then of the vase. This is all a big vase that maybe would have some flowers in it on like some wealthy person's porch or something, right? So this whole thing is the vase. Now it looks like Minnesota. But anyway, or let me take that away. Or if you were kind of focusing in more on the white, you might say, well, that's two people looking at each other, right? You're seeing these kind of profiles of people looking at each other. So the difference in what you see when I ask you what this is a picture of is a result of how your brain is actually processing and perceiving that visual information. all of us are getting the same exact raw data to V1, or our primary visual area. We're seeing the same shapes of white and black, but the way you interpret it, the way you perceive it, is going to be different. Here's another example. Think about what you're seeing. What is this a picture of? A lot of people will say this is a duck when they first see it. And you can see, absolutely, you can see there's the bill, there's the eye, you can see there's its little body, and then its little wing at the side, and its little feet. But there's another way to interpret it as well. You could also see that this is actually a bunny rabbit. So if you kind of change your perception a little bit, look at it from a different perspective, and what was the bill of the duck's beak, those are now the bunny ears, and the bunny is now kind of looking up in this direction, yes? And so it's facing the other way, and the rest is its body. So again, this is how you process that raw data that comes into V1. So this is done by other regions of the brain. These are called visual association areas. So these are the areas of our brain that are going to make sense of the raw data that V1 received. So in this diagram here, right, you've got your primary visual cortex, which is just the awareness of the visual stimuli. I've got this light over here and that light over there, this color here, this shape over there. And then we have all this visual association area, which is going to process this information to make sense out of it. There is something called the dual stream hypothesis. So in that last diagram, it was showing you areas of the brain that are all in the occipital cortex. But it turns out that when we are processing visual information, we're also using a lot of other parts of our brain. So the dual stream, is talking about a dorsal stream, which is on the dorsal side of the brain, or the posterior side of the brain. Again, think of dorsal fin. So this dorsal stream is where then we are checking in with some other areas of the brain to help us get a concept of where an object is. So what is its location? Is it moving, or is it still? The dorsal stream seems to be important for that. And then the ventral stream, which is coming down, and look at this, going right into the temporal lobe, here, is more of the what. So in order to figure out what something is, we compare it to our memories of other things, right? If I see something with two wheels and a seat in between and some handlebars, I might feel like, that's a bicycle. And I know it's a bicycle because I've seen a bicycle before. So we're kind of comparing that current raw data with our memories of other things that we've seen before and our knowledge about those objects that we've seen before. So that is the ventral stream, or the what is it that I'm seeing. So there are some specific examples of different areas of the brain that are part of this ventral stream. One is the fusiform face area, and that is the part of our brain that allows us to recognize faces. And believe it or not, there's a rare condition where people have an abnormality in that part of their brain and they can't recognize faces, even of their best friend, right? They couldn't pick them out in a room full of people because they just don't have that ability to process the information in that way. There's also another place called the parahippocampal place area, which helps you recognize places, buildings, and scenery and surroundings. So you can be like, oh, I know where I am. I'm in Jarvis Hall Science Wing, or whatever it might be, so that you can kind of get yourself oriented. So these are just a couple examples of how perception occurs, right? So we have all these visual association areas. We're checking in with other parts of our brain. We're checking in with memories. We're checking in with other information that's stored or that's coming in other parts of our brain. And it's super duper complex. So if you remember the whole white dress or blue dress controversy from several years ago, I don't know, maybe you were too young. Interestingly, when that first happened, it like stumped neuroscientists. They weren't sure why some people saw the dress as one color and other people saw it as another color. And then they got a whole bunch of data and they kind of figured it out. And that's what that link is about, if you're curious. So visual perception, how we perceive what that sensory information is coming in is extraordinarily complex. And what's interesting is it can also be tricked. So this is one of my favorite things. Sidewalk art, right? So these kind of visual illusions, for example. So someone has done a really great job of drawing on the sidewalk with chalk to make it look like there's this huge chasm that you have to jump across. And so we can trick our visual association areas by making it look like that's what is here. Here's another example of a way to trick your visual association area. I wouldn't want to stay in this hotel, right, and walk down this hallway. So the hallway floor is actually completely level and solid, but the pattern of the lines makes it look like there's, humps and divots and little craters in it, but it's actually fine. It's just the pattern of the lines that gives you that impression. This is another one. You could buy one of these rugs for your home. There's a great video, I couldn't find it to put in this lecture, sorry, of like a cat freaking out when it kind of walked onto this part of a rug like this. So other animals can be tricked too. This one is really interesting. I hope my head in the way doesn't mess it up. So this is a really interesting one because if you look at different areas of this picture, it will seem like some of the other areas, like in your peripheral vision, are moving. It's very psychedelic and crazy. So I'm just doing it a little bit, right? So obviously that's messing with our dorsal stream, right? That tells us about kind of position and movement. Fun stuff. So just remember, actual perception of vision is complicated and there are lots of pathways involved. Before we close out perception, I want to finish with the fact that some of the visual data isn't perceived at all. So we have some axons that are coming from the retinal cells, from those retinal ganglion cells, traveling the axons through the optic nerve that don't go to the lateral geniculate nucleus of the thalamus and then back through the radiations to the occipital cortex like we talked about. They bypass that entirely, and so we are not consciously aware of these visual signals, of this visual information coming in, but it does some important things for us. The first example is some axons that go to what's called the pretectal area. What is the pretectal area? It is part of the midbrain. So this is part of the midbrain, and you can see they've got pretectal nucleus, which is located on each side right here and here. And I think you can maybe appreciate there's this little tiny line because there's a few axons coming directly here instead of going to the thalamus, and same thing on the other side. So we have some axons that instead of going to the thalamus, they go straight to the pretectal area in the midbrain. And these are important for our visual reflexes. So when something is moving in your peripheral vision, a reflexive action is to turn and look at it. So to move your head usually, as well as also move your eyeballs to look at it. Makes sense as kind of a survival mechanism, yes? It also is involved when you're watching something move. That's called smooth pursuit. And actually, if we had to rely on our brain to consciously be telling our extraocular muscles, okay, a little bit more, a little bit more, a little bit more, it would be jerky and it would not be fast enough. So we actually use these reflexes to help us be able to follow a rapidly moving object pretty smoothly with our eyes and or our head as well. The second one that I want you to know about is called the retinohypothalamic tract. And it's called that because it's going to start at the retina, because this is visual information that's coming in. And then it's going to go through the hypothalamus, but then it's not done. So I wish that this was named a little bit differently, but let's take a look at it. So this is specifically giving us information about whether it is daytime kind of light, like bright and kind of blue wavelengths of light, which are what we normally see during the daytime, or is it nighttime evening kind of light, which is very much softer and dimmer and more kind of the red wavelengths of light. And so that's going to travel from our retinal ganglion cell along its axon through the optic nerve, but then it's going to go to this suprachiasmatic nucleus. Suprachiasmatic because it's above the chiasm, okay, which is located in the hypothalamus. So that's where the hypothalamus comes in. So that suprachiasmatic nucleus in the hypothalamus, and it's going to synapse there, and then we're going to send another signal to the pineal gland. And you might remember that the pineal gland is in that part of the brain called the epithalamus, also part of the diencephalon, and that is the part of our brain that produces melatonin. So in response to that dimmer, more red wavelengths of light, we produce melatonin, and that makes us sleepy. And so this is really important for our circadian rhythm and regulating day and night. So even if your occipital lobe was completely gone for some reason and you were not able to see or not able to consciously process any visual information, you would still actually follow a circadian rhythm, which is neat. So in summary, for perception, remember that just that raw data of the different colors and shapes are arriving at the primary visual cortex at V1, and it's regionalized, so different areas of the cortex are receiving information from different areas of our visual field, which also means different areas of the retina, because that all kind of maps out. So the word for that is retinotopic, right? So it's going to be different. But then we have these visual association areas, so V2 through V6, and other parts of the brain that are going to integrate this incoming information with other information that's coming in at the same time, as well as memories, things we've seen before, so that we can interpret and make sense out of what we're seeing. So remember that dorsal stream is kind of the where, and is it moving or is it still? And the ventral stream is more the what, comparing it to past memories. And then we talked about how some visual pathways we are not consciously aware of at all. So there's that pretectal one to the midbrain to help us with some visual orienting reflexes, as well as that smooth pursuit. And then the retinohypothalamic pathway, which goes to the suprachiasmatic nucleus of the hypothalamus. And then there's a synapse there, and then it goes to the pineal gland, which is then going to regulate whether or not we make melatonin to help with our circadian rhythm, our day-night cycle. So that is it for vision. I'll look forward to seeing you in lab.